I want to introduce Dr. Michael Wangler. He's an assistant professor and physician scientist at the Jan and Dan Duncan Neurological Research Institute and in the Department of Molecular Human Genetics at Baylor College of Medicine. He completed his MD at Baylor College of Medicine in 2006 and completed pediatric residency at TCH. He's board certified in pediatrics and medical genetics. His research interests include rare and undiagnosed disease and using genomics in medicine. He is an expert in HADDS syndrome as well as other genetic brain disorders. And we are very grateful for your participation and for your ability to do this talk for us. Thank you.
Thank you. Okay, thank you. And I guess people online probably can hear. Okay. So it's been a lot of information, and I will hopefully not add too much. My goal is actually to maybe re-emphasize some things that you heard in Haley and Tuan's discussion. So, and we sort of always divide this up a little bit where Dr. Chow focuses on the neurologic, and then I sort of focus on everything else, which is about proportionate. The brain is about, it's about right. So, so what I'm going to do is first I sort of want to go back to some of the things that Haley Streff talked about. And I want to talk to you about your DNA test results. So I'm going to sort of hopefully help break that down a little bit. And then we're going to go from that sort of genotype as a geneticist to the phenotype. So what's in your DNA versus what is happening, what are the traits, what are the disease manifestations and implications. So we're going to talk about the main features of HADDS from the medical literature. And then we're just going to talk about the process that we have of learning those features. And then I want to talk to you about the importance of doing this sort of for the future. Sorry. Okay, so many of you probably got a diagnosis of HADDS through a DNA test report originally. That may not always be true, but I would bet that most people got HADDS through a DNA test. And so basically this would be an example of the type of language that might have shown up for the geneticist or the neurologist that ordered. And you may have been given this, or they may have tried to explain it to you. I would actually encourage everyone. I know that many people participate in the research, and but I actually encourage everyone to obtain your own test report, not just like what not just your visit to the neurologist or geneticist, but you can ask for and you should obtain that piece of paper from the diagnostic lab. And it would basically probably look something like this. This would be an example, a heterozygous, and then it's going to sort of say, it's going to give coordinates. So this is the way that geneticists would spell in the genome. And then it's going to call it like a pathogenic variant or a likely pathogenic variant or a variant of uncertain significance. And then it may go into something about inheritance. So we're basically going to cover a lot of this in the following slides. You also might hear terms like novel, a novel variant. That would be like, we've never seen it before in the medical literature, so it's new to us as a medical community, or a variant of uncertain significance. So we know that there's a range of different types of mutations that can cause HADDS. So there would be missense, that would be changes that actually change just the spelling of the protein. Splicing, that would cause a problem with the RNA. And then there's indels, stops, frame shifts, those all interrupt the protein. And I'm going to talk about sort of the breakdown that we've observed for HADDS. This was some initial data when we were pretty early in the process. But you can see that, all of these essentially apply to HADDS. So many families will have different mutation than someone they meet with HADDS. All right, so another bit of nomenclature I just want to bring up is, so the phenotype, this is, as a geneticist, we think about, this is the outward characteristics or like the trait. So, a geneticist, you might think about traits that are not particularly medical, like height or hair color. But in this case, the trait would be like the presence of this condition that we sort of recognize the pattern of. And so that's going to be sort of what we go into in the second part is the phenotype. What are the characteristics of HADDS? And then there's the genotype. So that's kind of the DNA or the genetic constitution. And that for HADDS really comes down to EBF3. And in most cases, we're probably talking about what we would call point mutations, like small changes that are either a single letter change or a few letters changed within EBF3. There's some questions where I've realized that we have some individuals here with sort of more of a chromosomal deletion. So that would fall more under something like where there's an EBF3 deficiency, where the whole gene is missing. Maybe other genes in the region are missing. And then could also think about umbrella terms like EBF3 related disorders. So, as we sort of covered in Haley's talk, in all these cases, we still would describe the inheritance as autosomal dominant. So what does that mean? Autosomal just means it's on an autosome. It's not on one of the sex chromosomes. It's not on the X or Y chromosome. So in autosomal dominant, boys and girls would all be sort of affected in say a pedigree because it's not following the X chromosome, for example. And you could have inherited situations, as we've said. The majority of cases of HADDS are in this category where it's de novo. So that's still considered dominant, but it's different in that it sort of happens at a given time to an individual. And then, so we're going to cover a little bit because I'm sure that people have, or we hear questions that come up about this mosaicism, and we talked a little bit about it this morning. But I sort of want to spend some time on this issue of de novo changes and then where mosaicism fits in. One thing that I want to point out, though, that we did say before is that, if you're sort of over here, family A, or over here, family B, it's just important to realize that we don't really control how these processes work. So people worry about things that may have caused it. I'm going to talk a little bit more about what we know causes some of these things. But it's essentially, it has a lot more to do with chance than anything else. So this actually is showing, I'm going to go back to, you heard about Cole's kind of paper, but within that paper, we had to summarize all the data that we've collected so far. So this kind of shows the updated numbers for inheritance in HADDS syndrome for pathogenic EBF3 variants. And so what you can see is the breakdown is like 27 out of 41 are de novo. So the majority are de novo. And then, sort of 4 with affected mother, 1 with affected father, these four unknowns, you know, so the 27 out of 41 de novo, that actually may be an underestimate. In order to conclude that it's de novo, we would have had to get the DNA for both parents, and if we maybe can't get that, we may be missing some, and it's, you know, it's likely that unless we heard about something going on in one of the parents, that those are also de novo. And then we'll go into, there's some mosaic situations. So it came up this morning. I do want to say that there's two things to keep in mind about an individual with a mutation. So there were a good question this morning about that if a person has the same mutation as another individual, will they have the same, you know, sort of manifestation? And in general, it's sort of like we can see patterns, but no, I mean, they may be very different. Like the two patients, we would see, yes, they have HADDS, but one may have, you know, a certain area where like gastrointestinal that's strongly affected, and then the other one may have very little gastrointestinal manifestations. So that's called like variable expressivity is the term we use for that. And that's a very common thing that we see. That's different from the idea that somebody's walking around with no disease with the mutation. That we have not seen. So we believe that the mutations that we've described in this condition are all like fully penetrant. So they all cause HADDS. It's just whether sort of the flavor and severity would be the same is that's where the variation is. All right, so I like to talk about this because I think that for families where a de novo mutation has occurred, it's just something that, you know, it's natural to sort of think about it. So yes, I mean, it's true that radiation, like UV rays, X-rays, are mutagenic to DNA. There's also chemical mutagens, and you can consider like whether these show up in foods, at what levels, and these sorts of things. But from what from sort of the data that we have, DNA replication is like by far the biggest factor. So basically, you have to think about the problem. Like our genome has 3 billion base pairs. And then like if you think about the average sperm cell, it's going to be copying the 3 billion base pairs 50 times in a teenager, 1,000 times in a 40-year-old male. So that means that there's a lot of opportunity for simple copying error, and that's like overwhelmingly the cause of de novo mutations. And and honestly, that, you know, this is unfortunately just something that's largely by chance. And you can observe that every individual in this room has a certain number of like coding de novo mutations. The question is sort of where, right? So in the case of some individuals, it's within EBF3. In other individuals, it may not be as sort of medically significant. So it's really a matter of random chance. And so in general, we don't tend to suspect like environmental causes or you know, other kind of factors. So like the analogy I give is kind of a ridiculous picture, but like you know, so in like in antiquity, right? There were sort of these monks, and you would copy the great texts like sort of over and over, right? And so there were instances where like an entire sort of country had this misspelling in these ancient texts because like one monk early on made a misspelling change, and then all these texts were copied afterwards. And so, you know, the thing I try to say is, imagine your boss sort of gave you a book, it had 3 billion letters, and they say, you know, take this to the copy machine and copy it. And then, oh, by the way, like take the copy and copy that and do that 50 times, 1,000 times, you're going to have some errors. And it's sort of no one's fault really. It's just a feature of that copying process. So so the main factor though would be parental age. So for things like EBF3 deletions, rearrangements, chromosomal abnormalities, we actually would pay attention to the maternal age because that's where chromosomal abnormality risk is higher. And then for the point mutations, we would pay attention to paternal age, for the same reasons that I said. Okay. And then I just want to go into mosaicism. So we actually had questions that sort of covered all this morning fairly nicely. But so germline mosaicism, that's where basically we did testing. It looks like your family has a de novo change. We're still going to talk to you about germline mosaicism because we can't go in and sort of biopsy the ovary, for instance. So we can't rule out that there would be some some duplication of cells that could have that change. And we would quote like 1% would be really the risk of that. Somatic mosaicism would be where, say, there's an individual diagnosed with HADDS and the parent has detected, we detect somatic mosaicism in the parent. And that would be the case where you would say, oh, well, that could be up to 50% risk. Now, like the important thing is that a person with mosaicism can still only pass on one copy. So you can't have a person with mosaicism who then has a mosaic child like that. You only get to pass one copy. So you're either going to pass the misspelling or the clean copy. That's all. That's your only possibilities. Okay, so that kind of were the things that I wanted to cover regarding genotypes. I'm going to move on to phenotype. I don't know if there's any questions about what I've covered. We can have questions at the end also. Okay, so let me go on to phenotype and maybe questions will percolate. So HADDS was named hypotonia, ataxia, delayed development. I think it's interesting because like those are key features and those are relevant features. But like we could see those features in a lot of other diseases. There are things about HADDS that are much more characteristic. There's nothing like when we see HADDS in clinic, there's like nothing like it. It's a very unique condition, and it's not because of HADDS. Like it's not because of hypotonia, ataxia, or delayed development. A lot of that comes from actually a lot of that has come from the conferences, from getting together, from families talking, from realizing, wow, that's interesting that all the kids have, not all, but many of the kids have this sort of itch, or yeah, when they were born they didn't cry. These are all kind of features that didn't make it into the name but help us form the pattern of the phenotype. So I would say that all this starts with identifying sort of the EBF3 variant, but then with collecting and with having family meetings, you really can see a lot of deeper similarities that emerge. So in the literature, I'd say the original descriptions were, and this included like some of our original paper as well, right? So we learned a lot as we went. So dysmorphic facial features that include like deep set eyes, low set ears, dysphagia, GI symptoms, GU abnormalities, global delay, ataxia, poor head control, and speech delay. These are sort of the key features that were originally described. So this was like our last conference at the 2019 HADDS conference. We had a chance to see a lot of you, a lot of the patients, and I'd say from that a lot of insights emerged. We were seeing more things about the facial features. We had this whole discussion about the itch behind the neck, the newborn that was very quiet, the pain and sensitivity. Strabismus of the eye was like a very common feature. So we learned a lot, and so I would really encourage you to read Cole's paper. It's in medicalese. It's got like differential equations. It's got fruit flies. It has everything. You may not understand all of it, but it started with sort of carefully taking all the things that we learned starting at the HADDS conference, but then actually analyzing it in a lot more detail. And some things I'll point out that were covered a little bit before, but so we're able to observe kind of as you'd expect for an autosome. We can see females and males. There's the age range of the patients that were ascertained. We're picking it up more in the pediatric population. It affects all ethnicities. And then actually, one thing we noticed was that it can affect the growth parameters. It's not a strong effect, but you can sort of see that the patients over time may sort of lose a little bit in terms of height and weight, but that the head size will remain normal. And then, patterns have really emerged in non-neurological features that are very characteristic of HADDS. There's craniofacial, GI, eye problems, GU, and musculoskeletal. Here we actually tallied up a number of these features. So if you go down the craniofacial, the sort of prominent forehead, ear abnormality, lip anomaly, straight eyebrows, these are sort of the most common. In GI, constipation is pretty common. Hearing about feeding problems of some kind is pretty common, and GERD. And then, take a look at sort of strabismus and nystagmus. I mean, that's a pretty common ophthalmologic feature. And then GU findings are also like of great interest. And in fact, the next talk, Dr. Stanisel will go into a lot more detail, but here we sort of were able to tally up all the different manifestations. So, some additional sort of craniofacial features that we look at in the clinic, deep set eyes. So when you come to clinic, I often might make you do these measurements. There's also kind of a prominent forehead and a prominent jaw. So all these things are they're not, pathologic, but they just help us recognize a pattern of a condition. So the last point I want to make is why we need to do this. Because, it's sort of like, I've heard before kind of like, well, it's interesting, but how does it help our family? How does it help our child? And, I think the main points to make are as we go, we've been able to refine diagnosis. I think we started out with sort of hypotonia, ataxia, delayed development, and EBF3 mutations. Now you can see we have a 10-question scoring diagnostic scale, so it really does help us with diagnosis. It helps us compare it to other rare diseases. But I think the main point that I would make is that, we don't have therapies that are, currently ready But there's reason to think that things like gene therapy and oligotherapies are coming for many genetic disorders, and one barrier for any rare disease is really getting down the natural history, because the struggle that you're going to have is not just the technology of getting the therapy, but proving that it works. And even the FDA will look at, are you going to be able to prove that this works? And something that they're going to look at is, do you understand this disease? Like, do you really understand the natural history? You have a pediatric population. Do you know what adults look like? And so that they really will be asking these questions. The more organized that we are as researchers and the more integrated we are with you as families, the better that can go for us, because it's unlike sort of diseases where there may be many, many people and we could quickly get an idea of natural history. For rare disease, we really want to start on this, early. And then that also helps us, hearing from you, what are the things that are most difficult for you as a family. That helps us know where to focus energy for therapeutics. So that's kind of the reason why we look at these things in the clinic, the reason why we hope you'd participate in this study. So that's all I had prepared. I would just acknowledge the growing HADDS team here at the NRI, and then definitely, all of you for hosting us and having me, Ashley and Mallory and Yvette and the whole board. Do you guys have any questions? Yes.
So, do you think that the weight gain is less because of GI issues or something that the body is processing differently?
Yeah, it's a good question. I don't know for sure. I do think GI issues were kind of where I was going, because at birth it seems like the parameters are more balanced, where it's pretty uniform across your head size, your height, and your weight. And like, we know that, if you aren't getting adequate nutrition, not at the level of malnourishment, but just, sort of something like if you have pretty bad GERD or something, you could see an effect on like weight first, and that would not affect head size until much later. Yes.
Somebody else was ready. Have you seen many of the kids yet who are nonverbal be able to openly communicate at some point, or we haven't had an opportunity to see that yet?
I mean, it depends on the age when we would be seeing them. I think it's common. So I think for a while we were tracking in clinic the timing of the first word, and it was fairly common for that to be delayed from the kind of what the pediatric textbooks
Well,
defined.
our Ryan was about 18 months old when she got diagnosed. And then she probably didn't say her first word when she said mama till at least 4. She jabbers, babbles, she joins in the conversation like, why aren't you talking to me? Kind of thing. But she probably only has formed maybe 5 to 6 words so far.
At
She
age
understands them.
4.
Yeah. And what about the walking? If they walk, they can use the walker. Have you seen many walk for
Yes.
an extended period of time?
Yes, definitely. There's a couple patterns for walking. So there have been some patients that, sort of lost that ability, unfortunately. But that's
After.
but that wasn't that hasn't been like sort of the most common pattern.
Okay.
Actually, the most common pattern is that they walk.
Thank you.
Just sort of the most common.
Right.
But actually, I mean, it's another reason why. So like in Cole's paper, we're able to kind of see a signal of severity at that certain area of mutation type. But I think it emphasizes that the more that we would recruit you all,
Right.
and the more patients we can see, there may be other correlations that can emerge. So like this pattern of those individuals who sort of lose the walking. I mean, I think that's something we'd like to understand better, because that seems different, and it seems like maybe the genotype could affect it, but we need more, right? We need more data.
Okay. Super. Thank you.
Yeah.
It probably isn't going to be a question you're going to be able to answer off the bat, but just using your experience and your knowledge in the field. And it obviously depends on things like legislation and stuff like that. But when do you think something like gene therapy might actually be, a treatment that might be
Yeah.
available down the road? Like, are you thinking it's like 10 years, 20 years? I mean, obviously depends
Yeah.
on a whole bunch of stuff, but yeah.
Yeah. So I'd prefer not to give, because I actually think like if you told me you must guess a year, and I feel like I would be probably more likely to be wrong than someone who has no knowledge of the field. I don't know. Because like there's been therapies that have emerged for genetic disorders that if you had asked me the year before the therapy came, I would have said this will never be a treatable disorder. So but I would say like gene therapy is happening for like certain classes of disorders. Eye disorders, there's success. Blood disorders, there's success. CNS gene therapy is in trials for certain genetic disorders. So I think it's coming. Then there's also kind of like oligonucleotide therapy. Like there's other classes of sort of DNA, RNA therapy. It all depends on the genetic mechanism. Like if you think that this is because EBF3 function is gone and we have to give it back, or there's something abnormal about the mutation, and if we just got rid of that mutation, it would make things better. But all of these are things that have gone from sort of concept to where there are very real examples. They're small in number right now. But I think it's so it could be 10 years, but I would it it could be sooner. There could be barriers that make it that it's it would take longer. But I think my point is that the net like the one barrier could be, well, do you all have enough natural history data, right? That's one problem that actually comes up in rare disease right now. So I guess I would say there's just so many reasons for hope. Like gene therapy is increasingly becoming feasible in different disorders. Oligotherapies are working in rare cases. They're small molecules. So there's just a lot of avenues that we haven't had before. There's been things where I thought we'll never have anything for this disorder that we have now. So good. I thought I would get us back on time, but I put us